Okay, so this is photophosphorylation, which occurs in the chloroplast. So it also involves chemiosmosis. So where are my H plus and my reservoir? In this case here this is a thylakoid, okay, surrounded by thylakoid membrane, and there are a lot of Stalked particles in the membrane. So in the thylakoid space, so this is the thylakoid membrane, this is the thylakoid space, and then these are the Stalked particles, right? So this is a stroma, which is the fluid-filled space inside the chloroplast, okay? So my H plus are all stored inside the thylakoid space. So therefore, in the thylakoid space, it's got very high concentration of H plus. So it's going to diffuse through the Stalked particle, okay? So the neck region is the H plus channel protein, and the head region is the ATP synthase. So it's going to diffuse down its concentration gradient through the Stalked particle to a region where it's lower H plus concentration in the stroma. So when it diffuses across and turn this enzyme, ATP synthase, then ATP will be synthesized from ADP and PI. Okay, so therefore I can write here that ATP will be synthesized, okay, from ADP plus PI. It will form ATP. Okay, so the arrow will be like this. So, okay, and this is when my H plus pass through the Stalked particle and turn the enzyme. Okay, so my H plus. Okay, so this is chemiosmosis. So now I finished this story here about chemiosmosis. The next part is we'll talk about in a normal hydroelectric dam, when the water starts to flow out of the reservoir to the river, then the water will be recycled back to the dam, right, to the reservoir, by the water evaporating from the river, go up to the sky, become the cloud, and then will rain back to the reservoir. So in the cell, we also have to replace back the H plus in the thylakoid space, but it cannot rain inside the cell, right? Can rain or not? Maybe can, right? We don't know. So the cell cannot rain. So what is the way for us to recycle the H plus or bring the H plus back to the reservoir? Is we have to use this structure here called electron transport chain. Okay, so this structure here, electron transport chain, is the structure that will recycle the H plus. That means it will pump the H plus back to the reservoir. Whether it is the intermembrane space of mitochondria or the thylakoid space of the chloroplast, it's going to pump it back. Okay, so how does it pump it back? Is electron transport chain, right, is actually a series of electron carriers that you have learned last time. So let me draw up. So let's say, for example, this is the membrane. Okay, in the membrane, they got a lot of proteins, right? So the protein can be carrier protein, channel protein, electron transport chain, electron carriers, enzymes. Okay, so in this case here, they are the no, sorry, not square, circle. So they are electron carriers. So they are proteins which act like electron carriers. So these are the proteins that carry electrons. So I call all of them as the electron carrier. So this is the electron carrier here. So all these three are electron carriers. So when I label like this, I have to put this as an S already, electron carriers. Okay, to put it as an S here to be correct. So these are electron carriers. So what do? They carry electrons. So how do they carry electrons? So I've got electrons that come here, E, okay, and this E will be passed to the next electron carrier this way, okay, and then got an E here as well. So now I've got electrons already. Then it will be passed to the next electron carrier. Okay, So this is how they transport electrons versus, carrier protein, channel protein, they transport it across the membrane, right? Right, across the membrane. But this one, it transfers along the membrane, within the membrane. Okay, so this is an electron carrier. So this series of electron carriers all joined together, all of them are considered as the electron transport chain, ETC. Okay, ETC is a short form for electron transport chain. So what do they do? How do they transport the H plus back to the reservoir? Okay, so it's very interesting. So these electron carriers are protein, right? So every molecule has their own energy level, right? Every molecule got their own energy, right? Okay, so every carrier protein here got their own energy. So I represent the energy by drawing this bowl. Okay, I've got 3 bowls. So the first one is this big bowl here. Second one is a slightly smaller bowl. And the third one is even smaller. Okay, so the size of this bowl will represent the energy level of these 3 electron carrier molecules. Okay, so this one got higher energy, this one slightly lower, this one slightly lower. So for this bowl, let's say, for example, I want to transfer water from one bowl to another. So this bowl here initially can contain a lot of water, this volume of water. But when I want to pass the water from this bigger bowl to the medium-sized bowl, then there'll be some water that I cannot pass, isn't it? Because this bowl is smaller. So that means to say, when I pass the water from the bigger bowl to a medium bowl, there'll be some additional water that's left that cannot be transferred, isn't it? So this one is like when I pass electron from this electron carrier to the next one, because the next electron carrier got lower energy level, there'll be some energy that cannot be transferred to this electron carrier, right? Because the electron has got energy. The electron has got energy. Remember, your electrons are circulating around an element, right? There are electrons circulating an element, and you can add energy to electron, and electrons will go to higher energy level, right? So the energy of this electron can change, isn't it? It's still electrons, isn't it? So here, this electron has got energy, right? Has got a lot of energy. But when this electron is passed to the next electron carrier, the next electron carrier cannot hold so much energy of the electron. So therefore, some of the energy will be lost when this electron is passed from the first carrier to the next carrier, because this next carrier cannot hold so much energy. So there'll be excess energy that will be lost. So this energy will be lost in two forms. So this energy that's lost when electrons are being transferred, it can be lost in the form of heat energy. Okay. So this is where the heat from respiration comes from. It's lost as heat energy. And then some of them is enough. If it's enough, you can use to pump the proton. It's enough energy. Okay, so this is what happened. So it's like, okay, from a bigger bowl, I want to pass the water to a medium bowl. So there'll be some extra water that the medium bowl cannot hold. So this water I can make use of. Okay, I can make use of for other purposes. So when I pass the electron with a lot of energy to the next electron carrier, the next electron carrier cannot hold so much energy. So there'll be excess energy that can be released as heat. And if it's enough, it can pump the proton. So in the same way, from the second bowl, I want to pass the water to the third bowl. So the third bowl is a small bowl. It's smaller than the second one. So again, there'll be some water that cannot be transferred to the last bowl because it's smaller. So this water I can use for some other purposes. So in the same way, when I want to pass the electron from second to third electron carrier, the third electron carrier has got lower energy level. It cannot hold so much energy. So there'll be some excess energy, excess, some excess energy that I can use and lost as heat or to pump the proton. So therefore, every time an electron is being transferred along the ETC, some energy will be lost as heat as well as to pump the proton. So this is how it pumps the proton back to the reservoir. Okay? Can follow or not? Yeah, okay. Right, all good. So now continue. So let's recap. Chemiosmotic theory says that protons or H plus or hydrogen ions, they are stored in the reservoir. Now you cannot say reservoir in exam. You have to tell me exactly where. Because I use the word reservoir so that you can relate it to the hydroelectric dam. But you open all the textbook, they never say reservoir. It's for you to easily understand and remember only. So you need to say where. So in the mitochondria is in intermembrane space. And then in the chloroplast is in the thylakoid space. Okay. And then this proton will diffuse across the inner membrane or the thylakoid membrane because they got proton gradient. So there's a proton gradient, and this proton got electrical potential energy because it's charged. Okay, or electrochemical potential energy. So they have got the energy that they are able to move from region of higher to lower concentration. So this is their potential. Just like the water in the hydroelectric dam got potential energy, right? Because they are able to move downwards. So this one is able to move down concentration gradient. So it got potential energy. Okay. So when proton diffuse down their concentration gradient through the H plus channel protein of the Stalked particle into the matrix, then ATP synthetase is driven to synthesize ATP from ADP and PI. So can you see in the sentence, it's very crystal clear one. It tells you exactly what happens to H plus. It moves from where to where, and then what happens. Okay. So this will be your notes. All these are your notes here. All right. So and then protons will be pumped back to the reservoir by the electron transport chain, ETC. So ETC consists of a chain of electron carriers. So when electrons are passed from one electron carrier to the next, some energy will be lost. And the energy that's lost is used by the electron carrier to pump the proton through the membrane up the proton gradient. Okay. So this is the use of energy lost in ETC. It can be used. It can be released as heat, or it can be used to pump the protons. If it's enough, then we can pump. If it's not enough, we cannot pump. Okay. So this diagram shows you the y-axis is the energy level. So these are the 4 electron carrier proteins that arrange from the highest energy level to the lowest energy level in the ETC, electron transport chain. So I show you how when electrons are being transferred from one electron carrier to the next, energy will be released, and it can be lost as heat, or it can also be used to pump the proton. See? So when it's transferred, right, energy is released. Then transfer again, energy is released. Then transfer again, energy will also be released. Okay? So this is what happened in the ETC when electrons being transferred. When there's no electron being transferred, of course, no energy, right? There's no energy released. Okay? So this is another diagram to show you that, okay, so on the right side, you've got the Stalked particle. This is where chemiosmosis occurs. H plus pass through the membrane. Okay, ATP synthesized. Then on the left side, these are the electron carrier proteins, different size, different shape. All of them are considered as ETC. So this yellow color line shows you the movement of electrons. So when electrons are being transferred from one electron carrier to the next, can you see that now this electron carrier is pumping the proton, pumping the proton, pumping the proton. Okay. So this is the ETC act as the proton pumps when electrons are being transferred along the ETC. Okay. Now you don't have to read all the detail. All this 1, 2, 3, 4, the name no need to memorize. Okay. You need to know whatever I've mentioned here. All the details you no need to read. Don't see. No need to see. Okay. It's good to learn how to minimize things that you need to learn. Understand? Those that are not important, ignore. Okay. I'm recording, right? Okay. Cannot tell you more. Huh? Those people not important in life, ignore. Understand? Those people that when you always talk to, they're very negative and they pull you down, ignore. Those people who raise you up, okay, go osmosis more. You know osmosis? Just learn more from them. Get more positive chi from them. Understand? Then you also become more and more positive. Okay, Shamin. Charmaine is processing. Okay. All right. Okay. So what have you learned today is overall for this topic, this PowerPoint, cells need energy and where energy are being used for. Okay. We look at the structure of ATP. We see that ATP is a universal energy carrier from energy yielding reaction to energy requiring reaction. It's energy currency because you have to pay the cell to do the work. And then there are three ways that ATP can be synthesized. And all of them are called phosphorylation because we add Pi to the ADP. Okay, so you've got substrate level phosphorylation, oxidative phosphorylation, and photophosphorylation. Okay, so oxidative phosphorylation and photophosphorylation uses chemiosmotic theory. That means ATP synthesized by chemiosmosis. H plus pass through Stalked particle, turn ATP synthetase, and then ATP synthesized from ADP and Pi. Okay, so chemiosmosis is proposed by Mitchell, so it's called chemiosmotic theory. How ATP is synthesized by chemiosmosis is proposed by Mitchell, so it's called chemiosmotic theory. Okay, so what is chemiosmotic theory? I'll start from number 3. H plus diffuse across a Stalked particle, and then ATP synthesized from ATP synthetase, and then the ETC, when electron, point number 2, when electron flow along the ETC, proton will be pumped, okay, against constant gradient. So that will generate the proton gradient number 1. Okay, so all these 4 points are describing about the chemiosmosis. Is that okay for all of you? Okay, so I will continue with the next topic.